joining a group that was, very much interested in, development and morphogen and how you shape an organ. But then, things took a very interesting turn science-wise. So what was the challenge and how did it go?
Yeah, what attracted me about Steve's lab and science is during my undergrad time, I read the book, The Making of a Fly from Peter Lawrence. And this book, I think, is really kind of magic at least for me as a biologist, because I realized that complex phenomena, such as how do the bristles on a wing all grow in such a nice pattern and point to the right in the right orientation, that this can be explained by relatively logic molecular pathways, such in this case the notch pathway or planar polarity. And the fruit fly is really such an amazing model system to discover these principles. And to me, connecting biological phenomena to molecular principles sounded like such a fascinating topic. And there, Steve's lab with patterning and growth control was just a fantastic place to be. So I entered the lab, and my first project was to continue on the work of a grad student who was just about to leave. But that project, after half a year, didn't feel either. First of all, it didn't feel my own project because it was this continuation. And secondly, there were a few aspects that simply were not exactly as we initially thought. And I was a little frustrated. And then I decided, I don't exactly know why, but I decided to poke into one of the big mysteries in this group, which is, here is a locus in the fly genome. It's about 40 kilobases large, which for the fly genome is actually quite sizable. And we know something is in there, but we don't know what.
We can't
Some
find it.
gene must be in there, but we don't know what. And this puzzle was very attractive to me. I started to poke. Most people in the lab said, don't touch it. It will waste, it will ruin your career.
A lot of people tried.
Exactly.
Went nowhere.
Several postdocs already went down. And then I had to give journal club. If you think about journal clubs, many people have very different opinions about journal clubs because it's typically connected to some work, presenting, etc. Many journal clubs, I think, are also not terribly exciting. But here, it's a great example to show how valuable journal clubs are. I went to the library and started to look at journals. And I found this science issue where 3 papers reported the discovery of microRNAs in multiple different organisms. And when I read that, I kind of dreamed about maybe the Bantam locus, this magic locus that we didn't understand is a microRNA. This is how the project started.
You had a boss who clearly, left you freedom to do what you wanted. And the first question about that is how much you think this was a feature of the group or part of the culture at EMBL, and whether you still value that this is a good thing, actually.
Yeah, I thought about this question actually a few times. It's a very interesting question. So the project was centered on this locus, which Steve's lab identified. So I think Steve had a strong connection to this mystery locus called Bantam. And so from that perspective, I think for him it was kind of okay and natural to follow any work in this area. How did we make it, or how did we connect the dots, and what else was important? Essential for this entire project was, in fact, the collaboration with Alex Stark, a colleague of mine in the PhD program in the same year. And I think here are now a few aspects that are very interesting to consider. The only way for me to reach out to Alex was because I knew him from the PhD course. right? So the PhD course, this 3-month crash course at the very beginning where the cohort is together and gets to know each other, but also gets to know EMBL and all kinds of different facets of EMBL, enables these type of connections. I think PhD students are an ideal way to connect a place, to connect the labs horizontally. And so this happened here as well. What was really remarkable is that Alex's boss, Rob Russell, who was a protein structural biologist from the computational side, and where there was a clear agreement. Per Borg's lab is working on DNA and Rob Russell's lab works on proteins, that he allowed Alex to dive into nucleic acid biology completely outside the scope of the lab. I think that was kind of remarkable. And in retrospect, now I'm guiding also my own research group, you understand, I think, how much of a step that was.
Yeah.
Not normal.
So not freedom only at the student's level, but also freedom at any level.
I feel sometimes like a student because this field is teaching us all the time something new. And so that's what I love about the field. There's not only this one component. How does this piRNA system work? Again, this is small RNA based. These small RNAs are incorporated into an argonaute protein, and this argonaute protein finds complementary targets, in this case transposons, and is able to silence them. And so just the hardware of this pathway are, as I mentioned, about 30 to 40 proteins, and it keeps many careers busy. But it is connected to this evolutionary aspect of co-evolutionary aspect between transposable elements and host genomes. And that is a fascinating area of biology where as I said, I'm a student learning this field and all the different implications it has. For example, the vertebrate immune system would not exist without this conflict. So maybe to start, right? Transposons are selfish nucleic acids which have made it into a host genome, and they have the ability to multiply within this genome, in essence, at cost of the host. Because transposition, meaning amplification within a genome, is not only coming as a burden, you're increasing genome size, 50% of the human genome clearly traces back to transposable elements. It is also a mutagenic process, right? Because you might insert into a gene or an enhancer or disturb regulatory elements, etc. Nevertheless, this ancient genetic conflict, which probably dates back to the very beginning of genomes, has had fundamental impacts on biological innovation. And one example is the vertebrate immune system, because the key enzyme that is responsible for VDJ recombination, which is the process underlying antibody diversification, is an enzyme that we stole from a transposable element. Or you look at mammals, the placenta is an organ where cells fuse. And the only way how cells fuse is, cells don't fuse typically, right? Sure. And the way how they fuse is because these cells express a class of proteins called syncytins, and these are stolen from endogenous retroviruses, which are also considered transposable elements. So you see how this conflict is constantly shaping innovation in biology and evolution.